Okay, the next topic we want to talk about is genetic technology. So now that you really understand DNA, what it's made of, how it replicates, how do we take some of what we know about DNA and genes and how do we use it for some important technology? So that's what we want to talk about for a while. And you've seen what's shown in this diagram before. So you worked with plasmids in lab. We've talked about plasmids, which are these small circular pieces of DNA. And these are going to become important now when we start talking about gene cloning. So when we talk about any kind of genetic technology, we're really talking about manipulating DNA in vitro in some way. And I want to start by talking about gene cloning and sort of defining what that is and walking us through the steps of gene cloning. So there are a lot of words on this slide, in this diagram here, but let me just start by saying gene cloning. Another way we talk about it is recombinant DNA technology. We kind of worked through part of it in our molecular genetics lab that we did, and we're going to review and talk some more about some of those things, but recombinant DNA technology is really about taking DNA from two different sources and joining them together in vitro. And so you're going to see why that is important. And gene cloning is basically making lots of different copies of a particular gene. And you may want lots of copies of that DNA for a number of reasons, or you can also use gene cloning to make a lot of its protein product. So when you look at what's outlined here in this diagram, so again, as I just said, gene cloning is really, we're trying to achieve two main goals. We're trying to produce a large amount of a particular piece of DNA, a particular gene, or we're trying to produce a large amount of that encoded protein. And so let's first talk about how we clone genes and how we put recombinant DNA together. And then we'll talk about how we use those things for one of the two goals that are listed here on this slide. So this slide here outlines some of the first steps of gene cloning, and again, you've worked with and we've talked about some of the things that are involved here, but if we just take a look at this overview. So first of all, we need to work with some of the tools of gene cloning, and a lot of these tools, again, we're working in vitro to try to isolate and replicate lots of copies of the gene or have that gene produce lots of the protein that it encodes. And so in order to do that, one of the first tools we need are things called vectors. And plasmids, which I showed on the first slide, those and like the pGlow plasmid that you worked with in lab, those are examples of what we call vectors. And so these vectors carry the DNA into a cell, and they can actually replicate inside that cell. If we're going to make lots of copies of a gene, or that gene's going to make lots of protein, we need that DNA to be replicated in cells. And so plasmids are a classic vector. We can also use viruses as a vector because they replicate in cells as well. So we take plasmids, you can insert a particular piece of DNA or a gene of interest into that vector to generate a recombinant molecule, and then you introduce that recombinant molecule into a living cell. So again, this outlines some of those steps. And so we've talked about vectors, and now let's quickly review talking about restriction enzymes, which you guys visited in the lab that you had last week. So remember, restriction enzymes are these enzymes that recognize specific sequences along the DNA, and they cut the sugar phosphate backbone in two different places, one on each strand. And what you see in this diagram is an example of a restriction enzyme called EcoR1. You worked in lab with some others, but this is the sequence that's recognized by EcoR1. And you can see with the arrows there where it cuts. And when it cuts the two strands of DNA, it leaves these overhangs shown there on the bottom and labeled as sticky end. And when you have these sticky ends, so if we take a piece of DNA of interest and we cut it with a particular restriction enzyme, those sticky ends allow for complementary base pairing with another piece of DNA that's cut with the same enzyme. And we can see an example of that in the next slide, because remember what we're trying to do? We're trying to make a recombinant DNA molecule, clone this gene, and we want to then join our gene of interest with a vector. And in this case, we're joining our gene of interest with a plasmid vector. So we cut with a particular restriction enzyme, and then we can generate these sticky ends, which allow us to then join our gene of interest to our vector. So this shows a little more close-up of what we're talking about when we talk about these sticky ends. So I've got the DNA that was cut with EcoR1, and if I take that and incubate it then with my gene of interest that's cut with the same enzyme, those single-stranded overhangs there, the AATT on both of those, will hydrogen bond to one another. And then I can include the enzyme DNA ligase to actually join those gaps and form those phosphodiester bonds between those fragments. And so I can take my gene of interest, shown here in blue, and I can join it with my recombinant vector, which in this case is a plasmid, and in so doing I generate a new DNA molecule by joining a particular DNA from one source, my gene of interest, with another piece of DNA of another source. This allows us to then clone that gene. And so then the next step we need to do, once we've generated this new molecule in vitro, is we need to introduce it into some sort of a host cell. And these host cells can come in a variety of different types. So you guys in lab, we went through how to do what's called a transformation, and that is how you would introduce this recombinant molecule into bacterial cells. So let's look at that and then also talk about what we might use these things for. What kinds of things might this kind of genetic technology be useful for? So at the top of this diagram, I have my recombinant molecule. I've got my gene of interest attached to a vector. And in this case, if you remember when we looked at Piglo also had an ampicillin resistance gene on it. It had an origin of replication, and we've inserted now our gene of interest into this vector. And by the process of bacterial transformation, if you remember from last week, you can introduce this DNA then into bacterial cells and select for those cells that have the plasmid in this particular situation by growing those cells on selective media or cells that have ampicillin. And so we've taken this through the entire process. We've then cloned our gene of interest, and now that gene of interest can replicate in bacterial cells, and we can get lots of copies of that gene as we grow the bacteria. Or if we want instead to have lots of protein being made from that gene, we can engineer it in such a way that as we grow the bacteria, we get lots and lots of the protein that's being encoded by that gene. And you guys did this type of thing with the Piglo plasmid and saw the expression of the green fluorescent protein off of the plasmid Piglo that you worked with, like is shown in this next slide. So you couldn't probably see this as nicely and nice and green as in your lab handouts as we see here, but this is bacterial cells that are expressing green fluorescent protein. So the green fluorescent protein gene has been cloned, and now all of these bacterial cells are expressing this protein. So this is gene cloning. We've cloned this gene and we're making lots of that particular protein. And this is really interesting and fun to look at. But let's talk about some other ways where producing lots of copies of a particular protein might actually be quite useful. So when diabetes first came on the scene, and it was determined that insulin and giving people insulin helped with diabetes, initially insulin had to be purified, and often purified from the tissues of other animals, to be then cleaned and used as a medication. Well, with the advent of genetic technology and being able to clone the human insulin gene and then put that gene into bacterial cells and allow those bacterial cells then to produce insulin, the insulin that we use now is actually recombinant insulin, as they call it. And so you can see in the diagram on the right-hand side here, insulin is made up of a couple of different chains. There's an A and a B chain that's part of insulin. So you have both genes for the A and the B chains put into recombinant DNA molecules. Those molecules are introduced into bacteria. We grow lots and lots of those bacterial cells and purify those proteins then out of the cells. And together then in vitro, in a tube, we can make what we call synthetic or recombinant insulin. And that is how insulin is made and produced. And there are a number of other medications that are produced in this way. The blood clotting factor, factor VIII, that is used for the treatment of one type of hemophilia is used because we now have the gene cloned and we can express that gene and purify the protein. And also tissue plasminogen activator, or TPA, is another protein that is synthetically produced. This is a clot buster medication, and so it's commonly given to individuals who are undergoing some sort of stroke. And in the news lately, I want to bring in at the end here some other things that are of really importance. And so this also has to do with how the vaccines right now are being generated against the coronavirus. So in the next slide, I want to show you a little bit about that. So just to remind you, here in this slide, we see a picture again of the coronavirus, the one that we're all hiding from these days. And we do know the sequence, the genetic sequence of its entire genome. And you can see the genome there in the gray box on the far left. And there's one particular protein that scientists are interested in, and it's the blue one, that spike protein, or the SP, that's shown on the outside. And this is the protein that binds to the receptor on the human cells and allows for the virus to enter. And because it's on the outside, it's a really good target for a possible vaccine or a possible antibodies. People who have been infected with the virus generate antibodies. So you see the protein domain of the S protein on the bottom left there. And so what scientists have done is they take this gene that encodes the S protein, you make a recombinant molecule, you put it into a vector, you express that protein in bacteria, and so you get lots of spike protein being made. Here you also see some M protein that's being made. And then we take that spike protein and we inject it into animals and get those animals to generate an immune response to that spike protein, and possibly then those antibodies can block the infection of the virus. And these are the antibodies, too, when we talk about serological testing of people who have been infected with the virus. Generally, what we're looking for are antibodies that have been raised against those spike proteins. So the spike proteins can be generated in a recombinant fashion and injected then to mount an immune response to help scientists try to generate what's called a protein-based vaccine. And then let's look on the next slide to see how those antibodies might work to prevent infection of the coronavirus. So again, in this slide, we see the spike protein on the outside of the coronavirus, and we generate an immune response to recombinantly produced protein, hopefully that anti-spike protein antibody that you see on the far right can come in and block the ability of the virus to interact with the ACE2 or the ACE2 protein that's shown there on the surface of the cell. So you have the virus sitting on the surface of the cell, and if we can get some antibodies that would block that interaction against the spike protein, that is one way that scientists are trying to develop coronavirus vaccines right now. So this is how some of that genetic technology is actually being used. So we saw how we can get green fluorescent protein being expressed in bacteria. We saw that with the P-glow plasmid. But we know we can express proteins like green fluorescent protein or GFP in plants. We can express them in animals. And so I want to talk a little bit now about transgenic organisms. So transgenic organisms are organisms that carry genes from other organisms. And in the case of a lot of these transgenic organisms, the new gene, or the gene that's not normally part of the plant or normally part of the animal, is generally not carried on a plasmid like we were just talking about. It's often integrated into the genome. And so again, we can have transgenic plants. These plants here are expressing green fluorescent protein. And we'll talk some more about transgenic plants in a minute. But you can also generate transgenic animals. Here, obviously, these are mice, and we can see that some of them are expressing green fluorescent protein on their skin. So again, transgenic organisms, these are whole organisms, generally multicellular organisms, which of course bacteria are not, that carry genes from other organisms. So what kinds of things and how do we, what kinds of things might it be transgenic organisms be useful for, and how do we generate them? So let's go back and look again at some of the uses and how a transgenic plant is actually generated. So on the left here I have some examples of some transgenic plants, but again with transgenics we're putting genes in the plant cells that are from other organisms. And so we first isolate some DNA or clone some DNA. We make some recombinant DNA molecules you see in that top panel number 1 there. And in this case, we introduce that into a particular type of bacteria, and then that bacteria infects a plant cell, and that bacteria sort of injects its DNA into the plant cells, and so then that DNA becomes part of the plant genome. And we generate then transgenic plants. And so a couple of transgenic plants are often very helpful and useful as agricultural products. And there are 3 examples here that some of you may have heard of before. So you can nutritionally enhance certain plants, of course, and golden rice is an example of that. So normal rice is on the left there, white rice, and the yellow or the golden rice now expresses beta carotene, which is a form of vitamin A. And so in some parts of the world where rice is a big part of the diet, but certain vitamins are lacking in the diet, this has sort of been incorporated to give people a more nutritionally balanced diet. And then you see flavor saver tomatoes. These tomatoes are transgenic in that they express particular genes that make them more resistant to rot. And you can imagine as we transport tomatoes across the country, having rot-resistant tomatoes could be very important for that industry. And then at the bottom, some of you may have heard of BT corn. So BT corn is a transgenic form of corn that expresses pesticide, basically. So the corn borer, which is a pest, is actually killed if it tries to eat BT corn or corn expressing this toxin. So you can see on the bottom the BT corn is on the left and the non-BT corn is on the right. So this keeps farmers from having to spray a lot of pesticides on their fields because the corn itself carries a transgene for a particular pesticide. So these are some important agricultural advantages to making incorporating various genes into plants. And of course we can do the same thing with multicellular animals. And so transgenic animals, there are a number of uses for introducing other types of genes into animals. And here's one example where you would take, again, we've got in the top panel, in this particular case, we've cloned a human hormone gene into a vector, into a plasmid vector, and we've introduced then this human gene into a fertilized egg, and that DNA from the human hormone gene then becomes incorporated into the DNA of that sheep oocyte, and then that grows up to be a transgenic sheep. And you can, if you set this up correctly, that human hormone then can be expressed in the milk of this sheep and would be easily purified and used. And not only can you do this in sheep, you can do this in other milk producing animals because it's fairly easy then to purify the protein that's being produced from these animals. So this is sometimes referred to as molecular farming, generating some sort of pharmaceutical from a transgenic animal. And we also can use transgenic animals to help us treat disease, help us look at various animal models of disease, and we'll look at that in the next slide. So I'm not going to go into great detail on CRISPR-Cas9, how it was identified, where it came from. I mean, there's all kinds of information if you're interested. I put up a video. I'm going to put up a video on Canvas that talks a little bit more about this. But so we talk about transgenics, or altering the genome, changing the genetic makeup of an animal. And one way to do that is through very specific genome editing. And what CRISPR-Cas9 does is it allows us to go in and very specifically edit specific places and parts in the genome. And CRISPR-Cas9 really came into the scene very recently. We're talking about 2012. And it has just really taken the whole genetic technology field by storm. And so if we look at the panels on the left, this is a very oversimplified diagram of how CRISPR-Cas9 works. And so again, you're able with this technology to go in and literally you could add some sequence, you could remove some sequence, you can alter a sequence, and you can very specifically target where you want that change. So what you have, the top one there, is you have a target sequence somewhere in the DNA, in the genome of an organism, and you use what's called a guide RNA. And we can synthetically make these guide RNAs, and these guide RNAs would be synthesized with the particular change that we're trying to introduce. What is the edit we're trying to make here? Are we trying to remove a mutation? Are we trying to, or an alteration? Are we trying to add some new DNA? Are we trying to alter some part of the DNA? And you put that change that you want to implement, or that edit you're trying to make, you put that into your guide RNA. And then you add in a protein called Cas9. Cas9 is the big brown blob there. It recognizes this sort of RNA and DNA hybrid with my guide RNA. The Cas9 enzyme cuts the DNA at particular places in this whole complex. And then the guide RNA instructs the repair of that DNA. And it instructs the repair to maybe remove something if it's not on the guide RNA, to change it, to alter it in some way. So again, this is a very oversimplified diagram of how CRISPR-Cas9 works. But it's a very specific way to go in and edit or change the genome or the DNA of any organism. And there are many, many applications to CRISPR-Cas9, and you see a few of them in this circular diagram that I also have in this slide. And so we can, as shown with the various animals, we can use CRISPR-Cas9 to generate animal models, to go in and again specifically change the DNA to give us disease models and other types of things that we then can use to try to understand how diseases work, how medications for certain diseases work. You can use them to one day actually act as therapies in human disease. Can we go in and change the genomes of certain cells in humans? And actually, the first human treatment of CRISPR-Cas9 was done in 2019 in people with sickle cell anemia, to go in and try to genome edit out of their somatic cells the mutation that causes sickle cell anemia. And so you can see there are a number of other applications around this circle for agricultural purposes, food, other kinds of materials, going in and genome editing, making changes to various plants. What about, prenatal sort of things? Can we edit embryos? Other kinds of diagnostics. And so there's a whole slew. This is just a small bit of them, but there's a whole slew of applications for this genome editing process called CRISPR-Cas9, which is, again, a very specific way to target and change sequences in the genome. So up to this point, we've been talking about gene cloning. And again, remember, the purpose of gene cloning is to get many copies of DNA made over and over, or many copies of the proteins that are encoded by that DNA. And I wanted to just take a second to talk about the fact that we can also clone whole organisms. And this is, again, making a genetic copy of an entire multicellular organism. And this is actually really quite straightforward to do in plants. You can take a cell from pretty much any area of a plant, and if you grow it under the right conditions, you can grow a whole plant from that particular cell. But this was a much more challenging feat and had not been successfully accomplished in animals until 1996. And some of you may have heard of Dolly the sheep. And so the process of organism cloning from the DNA of a somatic cell is shown there on the right. But again, when I was with students this past January, we went to Edinburgh, Scotland, which is Dolly the sheep, and all of these experiments were carried out in Scotland. And Dolly the sheep has since passed on and died, but here she is taxidermied, and we were able to see Dolly the sheep at the National Museum of Scotland in Edinburgh, which was very exciting. So again, Dolly was the first mammal that was successfully cloned from the DNA of a somatic cell. So if we look at the top panel, so the DNA was taken from from the donor sheep, and you can see that that's the red nucleus there. And then that nucleus was fused into an egg, a donor egg, and then that egg was put into a surrogate ewe, and then that egg was able to develop into what we now know as Dolly. And so Dolly then was a genetic clone to the donor sheep, shown way back up in panel 1. And again, this took a number, I think Dolly was the one success out of over 200 attempts. But since Dolly the sheep in 1996, many mammals since then have been cloned. Mostly livestock, and you can imagine that there are some advantages to having a genetically homogeneous herd of cattle and various animals. And of course, we know in humans, identical twins are natural clones of one another. And there are companies too that allow people to do this same sort of thing, to clone their pets. And so, of course, there are all sorts of ethical considerations and other things to keep in mind with that. But not only can we and do we clone genes for lots of reasons, we can also now clone whole organisms. And this is the technique that's used to do that. And the last part I want to talk about here with genetic technology is a technique that you are familiar with since we did this in lab, and that is the polymerase chain reaction. And I'm only showing one cycle of that here. But if you remember the polymerase chain reaction, we have some template DNA, a number of primers, we put in some heat stable polymerase, and we put this through a number of different cycles. And so you can see here in the first cycle, the double-stranded DNA comes apart in a denaturation step at 95 degrees. And then we have the primer annealing step where the primers anneal to those single-stranded pieces of template DNA. And then you have the extension step where the polymerase then copies those pieces of DNA to give you two new double-stranded pieces. And then you continue this cycle. We go back to 95 degrees, back to the primer annealing and the primer extension for 30 plus cycles. And so the polymerase chain reaction allows you to replicate DNA. And now you know and understand better how DNA replication works. But it allows you to replicate DNA in vitro, right, in a test tube, without using any kind of vector or cell. You put in these reagents, and as long as you have everything working, you can amplify specific segments of the DNA without cells or without vectors. And this is what was done in our PCR genotyping lab that you guys did a few weeks ago. So just to remind you, again, you guys, we didn't do this unfortunately this year, but you had red and white-eyed fruit flies. We would have extracted the DNA from the flies, and then you performed the PCR and ran the gel electrophoresis, and it allows you then to determine the genotype of your fly. And so we see a couple of different genotypes shown there in the bottom of the gel. Lane number 1, you have the white-eyed female. Lane number 2, you have a red-eyed female, and then lane number 3, you have a heterozygous female. So you guys know you did this. You extracted DNA and used PCR to actually determine the genotype or the genetic makeup of your red and your white-eyed flies. And so you can extend this to determining the genotype of humans. So if you are, you're a carrier for a particular, or you suspect you're a carrier for a particular genetic disorder, you can be tested and to see if you are going to pass that on to your children. You don't know whether you're homozygous. You don't know whether you're heterozygous. You can use PCR to genotype and figure out what your particular genotype is. And so you can do these kinds of things prenatally as well. So let's look at the next slide to look at how that is done. So you can use PCR. Remember, PCR is just a really great way to amplify specific sequences from DNA that is in very small quantities. And so this is pre-implantation genetic diagnosis. So people who have certain genetic disorders that are segregating in their family, maybe they want to go through in vitro fertilization and make sure that they are the embryos that are being implanted are going to result in healthy offspring, and so they can genetically diagnose prior to implanting those embryos, and PCR is a part of this. And so you take DNA from a single cell. So early you do in vitro fertilization. You have a blastocyst, a very early embryo. You can take a single cell from the blastocyst, isolate the DNA from that single cell, and then carry out PCR and look at the PCR products to determine whether or not the embryo that cell came from, what's the genotype of that embryo. In this particular panel that's shown here, this is PGD, or pre-implantation genetic diagnosis for a disorder called thalassemia. And you can see in part D there The normal DNA has a T at a particular location, and the mutant DNA has an A. So you can amplify the DNA, you can determine the genetic sequence of that DNA, and with that determine the genotype of the embryo, and then make the decision as to whether or not you want that embryo to be implanted. So just as you can determine the genotypes of adults, and figure out, whether you're heterozygous or homozygous or a carrier for a certain disorder, you can also determine the genotype of an embryo before implanting it. Again, PCR allows you to do that easily because you can amplify a lot of DNA from a small amount of starting template DNA. A couple of other applications of PCR that I wanted to mention. So certainly we can use PCR in forensics. And what you're looking at on the left panel here are a number of bands that are generated through PCR. So you can imagine you could throw in, with your template DNA, you could throw in a whole bunch of different primer sets and actually amplify various regions around the genome all at once. And so that's what you're shown here, and then these bands indicate all of the amplified products. And you see the evidence lane on the far left, and then you see the PCR products from 3 different suspects. And I don't know what the forensic sample was here, but you can imagine you can do PCR by amplifying DNA extracted from maybe a small amount of blood at a particular crime scene. You can use sperm samples in the case of rape. You can just take the root of hair if there are hair samples left behind, and you can amplify the DNA from those small samples and compare then the PCR products that come from them. And we can see here when we look at the evidence sample, and I see that suspect 2 and the evidence sample match pretty well. And so that doesn't necessarily prove that suspect 2 committed whatever crime this is, but it certainly allows you to exclude suspects 1 and 3 from being considered as having committed the crime. So it's a very important tool to use in forensics. The other panel here is PCR, what we use with ancient DNA. And so what you're looking at here is bone being kind of ground up, and we can extract DNA from ancient bone. We can extract it from ancient fossilized spores from plants, and then use PCR to amplify some of that to get enough of that DNA to then determine its sequence. And it has really allowed us to study the genomes and the genes of some extinct species. And some of you may have heard a number of years ago we were able to get some sequence from DNA from the Neanderthal, and we're able then to go and compare that DNA sequence to modern humans. And this is how that those DNA sequences were obtained, was by extracting DNA from bone and then using PCR to amplify that DNA and then comparing that to the modern human DNA. And so I want to end this with something, again, having to do with what is going on right now with the coronavirus and how PCR is used for that. So we all have heard in the news, probably more than we'd like to, coronavirus testing. Well, one of the tests that they're talking about is a PCR test. And so this is showing you sort of an outline of how that works to detect virus. And again, you swab the nasal, you take a nasal swab, extract the RNA in this case, because the coronavirus that causes COVID-19, its genome is RNA. And to do PCR, you do PCR on DNA. You amplify DNA, not RNA. So you extract the RNA from the specimen and convert that to DNA, and then there are specific primers that are put into the reaction, and those primers recognize some specific sequences that are only part of the coronavirus, and it's set up so that if it does amplify those sequences, there's some fluorescence or some shining, glowing results that are given off, and if they are positive, you should see fluorescence. If it's negative, you don't see fluorescence. And so PCR is the technique that is used to determine whether or not an individual actually has viral genome, viral RNA in their nasal passages. So all of these genetic technology things that we're talking about are things that scientists are using right now to help us learn and understand and get through the pandemic. And PCR is used in the COVID-19 testing.